We're moving on to part 2 of our lecture regarding the structure and function of the cardiovascular and lymphatic systems. Now that we understand how blood flows through the heart with the pumping of the ventricles, we have to understand the conduction system of the heart. Let's first start with the SA node. And normally electrical impulses arise at this sinoatrial node. This sometimes is referred to as the pacemaker of the heart. And this SA node is located at the junction of the right atrium and the superior vena cava, just superior to the tricuspid valve. This node is heavily innervated by not only sympathetic nerve fibers, but also parasympathetic nerve fibers. The SA node generates about 60 to 100 action potentials every minute for an average adult at rest. It depends, though, on the age and the physical condition of that person. Now, each action potential will travel very quickly from cell to cell and through the atrial myocardium, carrying this action potential onward now to the AV node, or the atrioventricular node. This is going to cause both the atria to contract, which will begin the systolic cycle, or systole. The AV node that sits in the right atrial wall superior to the tricuspid valve and also anterior to the ostium of the coronary sinus conducts action potentials onward now to the ventricles, innervated by nerves from the autonomic parasympathetic ganglia, serving as receptors for the vagus nerve, and also causes slowing of impulse conduction through the AV node. Now we have conducting fibers from this AV node that converge to form the bundle of His, sometimes referred to as the atrioventricular bundle. Within the posterior border of the interventricular septum lies this bundle of His, and it gives rise to the right and left bundle branches. The right bundle branches, sometimes referred to as the RBB, is thin and travels within much branching to the right ventricular apex. And because of how thin it is and it relatively lacks branches, the RBB is very susceptible to interruption of impulse conduction by damage to the endocardium. The left bundle branch, or the LBB, this divides into two other branches, or fasciculus. The left anterior bundle branch, or the LABB, passes the left anterior papillary muscle right at the base of that left ventricle. It's then going to cross the aortic outflow tract. Whenever there's damage to the aortic valve or to the left ventricle, this can interrupt this left anterior bundle branch. This left posterior bundle branch travels posteriorly, crossing the left ventricular inflow tract to the base of the left posterior papillary muscle. This branch will spread then diffusely through the posterior inferior left ventricular wall. Blood flows through the portion of this left ventricle, relatively non-turbulent, because it's allowing the left bundle branch to somewhat protect itself from injury that's caused by the wear and tear. Now we're moving on to the Purkinje fibers, and these are the terminal branches of the right bundle branch and the left bundle branch. They extend from the ventricular apexes to the fibrous rings. They penetrate the heart wall to the outer myocardium. The first areas of the ventricles that now become excited are portions of the intraventricular septum. This septum is activated from both the right bundle branch and the left bundle branch. There's an extensive network of Purkinje fibers promoting this rapid spread of impulse to the ventricular apexes. This is going to allow activation. The basal and posterior portions of the ventricles are the last to be activated. We're going to start talking about some important concepts regarding the cardiac action potentials. There's electrical activation of our muscle cells. This is called depolarization. It is caused by the movement of ions like sodium, potassium, calcium, and chloride across the cardiac cell membrane. Deactivation is called repolarization, and it occurs in much of the same way. Now, movements of these ions or electrolytes into and out of the cell creates an electrical voltage difference across the cell membrane. This is called the membrane potential. And during depolarization, which is when we have electrical activation, the inside of the cell is now going to become less negatively charged because we're trying to move positive ions inside. In cardiac cells, as in other excitable cells, when the resting membrane potential becomes less negative with depolarization, reaches a threshold potential for cardiac cells, the cardiac action potential can now be fired. And there are various phases of this cardiac action potential that's related to the changes in the permeability of the cell membrane to these ions, to sodium, potassium, chloride, and calcium. Threshold is the point at which the cell's membrane's selective permeability to these ions is temporarily disrupted, and this leads to an all or nothing type depolarization. Now, drugs that alter the movement of these ions, for example, calcium, have profound effects on the action potential and can alter the heart rate. If the resting membrane potential becomes more negative because of a decrease in the extracellular potassium concentration, or if the patient becomes hypokalemic, this is termed hyperpolarization. Refractory periods are times when no cardiac action potential can be initiated by a normal stimulus following depolarization. Abnormal refractory periods are a result of disease, which can cause abnormal heart rhythms, or sometimes we call these arrhythmias or dysrhythmias. Think about ventricular fibrillation or even cardiac arrest. An electrocardiogram can show this electrical activity. ECGs originates from the myocardial cell electrical activity as it's recorded by electrodes on the skin, and it is a summation of all the cardiac action potentials. First, we'll look at the P wave, which represents atrial depolarization. The PR interval measures time from the onset of atrial activation to the onset of ventricular activation. And this interval represents the time necessary for electrical activity to move from the sinus node through the atrium, through the AV node, the bundle of His, Purkinje fibers, which activate ventricular myocardial cells. The QRS complex represents the sum of all ventricular cell depolarization. And the configuration and amplitude of the QRS complex may vary considerably among individuals. Now, during the ST interval, the entire ventricular myocardium is depolarized. The QT interval is sometimes called the electrical systole of the ventricles, but the time it takes varies inversely with our heart rate. And finally, the T wave represents ventricular repolarization. You will get an in-depth full course on ECGs. It's important for us to understand the autonomic nervous system, the sympathetic and parasympathetic nerves as they relate to cardiac innervation. The autonomic neural transmission produces changes in the heart and circulatory system faster than metabolic or humoral agents will. The speed is important. Let me give you an example. In stimulating the heart to increase the pumping action, which is needed with increased physical activity, or even during times of stress and fear, or the fight or flight response. And although increased delivery of oxygen, glucose, hormones, and other bloodborne factors sustains this increased cardiac activity, that rapid initiation of increased activity is going to depend on the sympathetic and parasympathetic fibers within the autonomic nervous system. The sympathetic and parasympathetic nerve fibers innervate all parts of the atria and the ventricles, as well as the SA and the AV nodes. So generally speaking, the sympathetic stimulation increases electrical conductivity and the strength of the myocardial contraction. It's also responsible for the vagal parasympathetic nerve activity, which does the opposite. It slows the conduction of the action potentials through the heart, reduces the strength of contraction, and this is why the sympathetic and parasympathetic nerves can affect the speed of the cardiac cycle, affecting the heart rate, or what we term beats per minute. The sympathetic nervous activity enhances myocardial performance. Stimulation of this SA node by the sympathetic nervous system rapidly will increase the heart. This system, the sympathetic nervous system, also can induce an increased influx of calcium, which will then increase the contractile strength of the heart. Also, the speed of electrical impulses through the heart, muscle, and the nodes. The sympathetic nerve influences the diameter of the coronary vessels as well. Increased sympathetic discharge dilates the coronary vessels, causing the release of vasodilating metabolites, resulting from increased myocardial contraction. The myocardial cell is nearly identical to skeletal muscle cells, but let's look at some of the major differences. Now, cardiomyocytes are long, narrow fibers that contain bundles of longitudinal myofibers. It has a nucleus, mitochondria, internal membrane system, which we call the sarcoplasmic reticulum. It has a cytoplasm, referred to as the sarcoplasm, and a plasma membrane called the sarcolemma. This all encloses the cell. Cardiac and skeletal muscle cells also have an external membrane system made up of transverse tubules or T-tubules, and this is formed by the inward pouching of the sarcolemma, remembering that the sarcolemma is the plasma membrane. The sarcoplasmic reticulum forms a network of chambers that surround the muscle fiber. Now, because the myofibrils in both cardiac and skeletal muscles consist of alternating light and dark bands of protein, they both will appear striated or striped. And the dark and light bands of the myofibrils create this repeating longitudinal unit, which we refer to as a sarcomere. Sarcomeres measure between 1.6 and 2.2 micrometers long, and the length of these sarcomeres determines the limitation of the myocardial stretch at the end of diastole, and then subsequently the force of contraction during systole. Whenever there is alterations in sarcomere size that are seen, this can be both a physiological issue or maybe a pathologic myocardial hypertrophy that limits the size or the stretch. There's also a number of differences between cardiac and muscle cells in terms of their arrangement. Cardiac cells are arranged in branching networks throughout the myocardium, but skeletal muscle cells tend to be arranged in parallel units through the length of the muscle. Cardiac fibers only have one nucleus, whereas skeletal muscle cells have many nuclei. Let's look at the differences between the cardiac and skeletal muscles relating to the heart function. The heart myocardial cell transmits action potentials quickly from cell to cell, and there's electrical impulses that rapidly move from cardiac fiber to cardiac fiber because there's a network of intercalculated discs, and these are thickened portions of the sarcolemma. The intercalculated discs contain three junctions. We have the desmosomes. Sometimes we call them the macula adherens. We also have the fascia adherens, which is mechanically attached to one cell and then to another. And then we have gap junctions, which allow the electrical impulses to spread from cell to cell. Any changes in the function of these junctional elements can cause an increased risk of arrhythmias. The second function is to maintain high levels of energy synthesis. And unlike skeletal muscles, the heart can't rest and is in constant need of energy, which is supplied by molecules such as ATP or adenosine triphosphate. The cytoplasm surrounding the bundles of myofibrils in each cardiomyocyte contain a large number of mitochondria. Cardiac muscle cells have more mitochondria than skeletal muscles. And lastly, they're to gain access to more ions, particularly sodium and potassium. Cardiac fibers contain more T-tubules than muscle skeletal fibers, and also there's an increased closeness to the T-tubules, which gives each myofibril in the myocardium faster access to molecules that are needed for transmission of action potentials. And because the T-tubule system is continuous with the extracellular space and the interstitial fluid, it facilitates this rapid transmission of the electrical impulses from the surface of the sarcolemma to the myofibers inside the fiber itself. And this rapid transmission is activating the myofibrils of one fiber simultaneously. Myocardial contractility is a change in the tension at a given resting fiber length, which basically is describing the ability of the heart muscle to shorten. And within each sarcomere, there's a basic contractile unit of a muscle cell. That outward-facing heads of the myosin molecules are called cross bridges because they can form force-generating bridges by binding with exposed actin molecules. Once they're bound together, the myosin molecules will then pull the thin filaments toward the center of the sarcomere, shortening the length, which will result in contraction. This is what we call the cross-bridge theory of muscle contraction. And the degree of shortening is going to depend on the amount of overlap between the thick and the thin filaments. We also need to understand the excitation-contraction coupling, which is a process by which an action potential arrives at the muscle fiber plasma membrane, which triggers a cycle. It leads to the cross-bridge formation and contraction. And the cycle activation depends on calcium availability, and the amount of force that's developed is regulated by how much of the concentration of calcium ions increases within the cardiomyocytes. Calcium enters the myocardial cell from the interstitial fluid after electrical excitation takes place, which increases the membrane calcium permeability. And calcium entering the cell triggers the release of additional calcium from the two storage sites within the sarcomere. And then they diffuse toward the myofibrils, where they will then bind with troponin. Here is another opportunity to test your knowledge. Which of the following is true regarding the cardiac and skeletal muscle fibers? A. Cardiac fibers have fewer mitochondria than skeletal muscle fibers. B. Cardiac fibers are parallel and skeletal muscles are branched. C. Cardiac fibers have a slower transmission than skeletal muscle fibers. Or D. Cardiac fibers only have one nucleus and skeletal muscle fibers have many nuclei. Now's an opportunity to stop and review your slides before answering. If you answered D, you're correct. Cardiac fibers do indeed have only one nucleus. The fibers are branched, where skeletal muscles are parallel. Cardiac fibers have rapid transmission and abundant mitochondria. They have abundant mitochondria because cardiac muscles cannot rest, and therefore they need a superabundance of mitochondria because they need constant energy supply. Thank you for listening to the part 2 of our presentation. Please join me in the next lecture for part 3.